important, and we do a lot of it. So it's definitely something worthy of study, and I think it's surprising that, in fact, we really still don't know why we sleep. As he mentioned, a couple attractive hypotheses are that it's somehow restorative, and then the other possibility is that it plays an important role in memory consolidation. It's interesting that within maybe the last 5 years or so, there's really been an increase in the amount of research on sleep. I've really noticed this, a lot more talks now on sleep, a lot of people are doing research on sleep. Adults sleep on the average about 7.5 hours a night, which again means that you're sleeping about a third of your life. And the amount of time people actually spend sleeping goes down as you get older. The fact that sleep is so important, I think, is really demonstrated also by studies in rats, where the rats will sleep deprived. So in these experiments, they took a couple of rats and they had them use a device, and they had them both hooked up to EEGs, electroencephalographs, so they're recording brain activity. And you can, various stages of sleep have characteristic brain activity, so you can figure out, you can determine from the EEG when the animal's starting to go to sleep. So in this case, they had one animal, the experimental, that every time his brain waves started to look like sleep, they rotated this platform to keep him awake. And the other guy, they didn't do that. Apparently, he's sometimes being woken up by movement of the platform. Other times he's probably awake during movement of the platform. So his sleep isn't as disrupted. And they found that the animal that had disrupted sleep, that wasn't allowed to really sleep for a number of days, eventually, after 28 days, died because of lack of sleep, basically. So you can see how important sleep is. We all have circadian clocks. So this is a clock that's wholly contained within us, and it keeps time. And so if you want to look at a circadian clock, much like when we were talking about central pattern generators, if you want to see that you have a circuit that's generating a pattern, or if you want to see that if you have part of your brain generating a clock, the way you look at this is to eliminate all sensory input, right? And see if there's some endogenous pattern on the clock. And so this is the case with, we look at, so this probably represents locomotive behavior. Usually that's what they're making. I'm not sure what exactly you're looking at. This is with a white dark zone. And this is without any cues. All right. And so the line represents the portion during the day when you're awake or active or moving around or whatever. And you can see that, in fact, once you get rid of all of the cues, you still have a cycle. You're awake for a certain part of the day. Now, one thing you can see here is that it shifts every day to a little bit later, which is typically the case for these free-running circadian clocks. Left on their own, without any cues, they actually usually run at duration slightly more than 24 hours. So that, in fact, if they're free running, they start a little later every day. Now, typically, obviously, we get cues, usually light, which entrain the clock. and keep it so that it's basically starting about the same time every day. This entrainment of the clock can be produced in many ways, but the most typical way is by light. And the cells, the receptors, that are the most important for training the clock, and you might expect, are in your eye. Surprisingly, they're not your photoreceptors. There are actually some retinal ganglion cells in the retina which have a photopigment. Melanopsin. So these are light-sensitive ganglion cells. We haven't talked about these at all. So far in the retina, all we've talked about are the photoreceptors that are being said that are sensitive to light. These are actually sensitive to light. And unlike the photoreceptors, when you shine light on them, they depolarize and fire action. Remember, the photoreceptors, when you shine light on them, they hydropolarize. Obviously, so these depolarize, they fire action potentials, and they send action potentials rather than the optic nerve. These axons for entrainment project. One area where they project is the suprachiasmatic nucleus, the SCN. And it's the SCN which is thought to be our clock. Most of the clock functions are a result of the SCN. Receives input for entrainment from the eye. It has output to many regions of the brain. One area pointed out here is the pineal gland, and the pineal gland releases melatonin, which is thought to be important in sleep. How does the clock work? Okay, that's been a big area of interest for a long time. And requires a molecular description. You need to know at the molecular level how the clock works. And this whole field of the molecular mechanisms of the clock began in 1971 with the identification of the first clock gene. So these studies came out of Seymour Metzger's lab, Caltech. And if you remember, about this same year, it was either 71 or 72, that the first learning gene was discovered. Again, out of Seymour Metzger's. The Dunce gene came out of Seymour Metzger's lab about the same time. So it was a very important lab in study of their genetic basis of behavior. And of course, these studies were all done with Drosophila. So a typical approach has been over the last 30 or 40 years to find these genes in Drosophila and then to go look at them in mammals. And the PER gene is discovered in Drosophila. It's also found in the brain in the SC. So the approach is that you mutagenize the flies. You give them a drug, ethyl methane sulfate, not that important, it's usually referred to as EMS. And this causes point mutations in the genome. And you take the male flies, which will have some sort of random point mutation in their germline. And then you go through a series of crosses. So you get progeny at some point, in which you're homozygous for that gene, or sorry, for that point mutation. Okay. When you take these flies, you have this random point mutation. And you need to be homozygous because these mutations are often recessive, right? So you wouldn't see them if you were heterozygous. So that's why you have to go through a series of process. You take these flies in, and then you have some sort of behavioral screen. And this has been done thousands, hundreds of thousands of times now for various behaviors to look at the genetic basis. In this case, what they did was they took these flies and they looked at their circadian rhythms. So they're looking for a gene mutation that affects circadian rhythms. And obviously, since these mutations are random, you know, you have to look at thousands and thousands of flies. So you need some sort of behavioral assay you can do very quickly. Or you need a lot of genetic students too. And it's usually a little bit of both. Okay, so these are locomotor behavior. Yeah, locomotor activity rhythms. And this is one day. These are similar to the ones I just showed you before. And this indicates when the animal's active. So presumably this represents a couple days. He's active here, he's sleeping, and now he's active again. Next day he's active, he's sleeping, he's active. And flies actually do sleep. I should point out that these are multiplying. That I really noticed in the fly community that there's a lot of research not made on this. And you know, it's a little hard to determine when a fly sleeps because they obviously don't close their eyes, right? You can't see their eyes are closed. And it's a little hard to hook them up to EEGs or something, and I'm not sure that would show anything anyway. So there's some criteria, if I remember right, that if the animal does not move for a period of like a minute or a minute, or two, that constitutes sleep. That point for sleep. No one really knows what flies think. They can't self-report anything. That's been taken into these. So they looked for mutants, and they found these mutants which had different periods. So these guys had a short period of sleep, of circadian rhythm, and these guys have a long period. And these various alleles all map to the same gene, and they called this the period gene. Which, in short, the PER gene, and anybody in the Drosophila field, or probably anybody who works with clocks, would know what the PER gene is. So they identified this gene, and then many years later, quite a few years later, this is 1990, they looked at the PER gene product. What's the protein produced by this gene? And they found that the amount of protein actually had a circadian rhythm. So if we, so these are studies, this is one region of the fly brain, and they did immunocytochemistry, so they generated an antibody to the PER protein, and then they stained for that protein using this antibody. And they found that at certain times, they got strong staining for the protein, certain times of the day, and other times of the day, they got weak staining for the protein. They quantified this, the relative staining intensity, or the amount of protein, and they found a circadian rhythm. So, this gene is important for the behavioral circadian rhythm, and the protein it produces also has a circadian rhythm. You can see here, at night it starts to go up, and then during the day it drops down. Now, since that time, there's some other clock genes identified, and people are putting together a rather complex, as you might expect, mechanism, molecular mechanism for the clock. And just to give you some appreciation of that, I want to show you a quick video. I don't expect you to know exactly how the clock works. We're just mainly focused on PER gene, just to show you that this area more complex than just the purging.